do Dr. Pedro Beltrão, do EMBL, do Reino Unido, né? Ele vai falar um pouco para a gente do seu trabalho relacionado a proteínas de fosforilação, a sua relevância, né, com doenças. Antes de começar mesmo, de passar a palavra para o João Paulo e para o Pedro, eu só gostaria de falar um pouco a respeito do evento. Então, como vocês podem ver, né, o BRAZWEBINAR é essa série de webinars onde você tem vários speakers influentes na área da bioinformática, tá? Ele foi, para quem não conhece, ele foi idealizado aí pelo professor Miguel Ortega, está sempre presente nesse nosso evento, e é um evento que foi criado com o objetivo de tentar conectar, né, Miguel, os diferentes programas de bioinformática no Brasil, dentro desse contexto aí do cenário pandêmico. E é uma iniciativa que, na minha opinião, tem sido um sucesso, né? Hoje é a 6ª edição do BRAZWEBINAR com o Pedro. Ele tem alcançado audiências de toda parte do Brasil e também de fora do Brasil. E temos também 8, na verdade, professores de 8 institutos federais acadêmicos, né, que estão contribuindo aí para trazer esses pesquisadores como o Pedro para poder compartilhar com a gente sobre os seus pensamentos, sobre as suas pesquisas aqui nesse nosso evento do BRAZWEBINAR, tá? Então, também quero parabenizar aí o Miguel por esse evento importante, tá? E só para lembrar a todos que o BRAZWEBINAR é um evento que tem um cronograma até o dezembro deste ano, então convido vocês também para checarem o cronograma das palestras que já estão confirmadas lá no site do BRAZWEBINARS. Basta colocar no Google que aparece na primeira página, tá certo? Convido vocês também para já se cadastrarem, né, preencherem o formulário do próximo evento do BRAZWEBINAR, que vai ser no dia 7 de maio, e compartilhar isso com os seus colegas que apreciam aí os tópicos nessa área da bioinformática, tá? Então, gostaria já também, Pedro, de agradecer você pelo convite, por ter aceito esse nosso convite, é um grande prazer estar aqui com você, tá? E vou passar a palavra para o professor João Paulo, que ele vai ser o chair dessa sessão e vai introduzir para a gente o Pedro, tá? Então, passo aí a palavra para você, João Paulo.
Muito obrigado, Tetsu. Eu vou apresentar o Pedro, né? Eu vou apresentar o Pedro em inglês e qualquer coisa, as perguntas podem ser colocadas em português também. Tá? Good day, everyone. I want to thank Dr. Pedro Beltrão for accepting our invitation to the BRAZWEBINAR, the info event. We hope you can visit us in the future. You can visit Brazil, our universities, and our research groups. Dr. Beltrão has a PhD in biology from the University of Aveiro, with the research conducted at the EMBL Heidelberg in 2007. After that, he was a postdoctoral researcher at the University of California, San Francisco. And since 2013, he is a group leader at EMBL, European Molecular Biology Laboratory and European Bioinformatics Institute, researching the cellular consequence of genetic variation. Beltrão groups studies how cellular functions have diverged during evolution and how they are altered in disease using post-translational modification and regulatory networks. Please, Pedro, feel free to start your presentation. And again, thank you very much for accepting our invitation.
Thank you. So I hope you can see the screen. Eu vou falar em inglês só porque é mais fácil, mas se quiserem fazer perguntas em português também podem. Thank you for the introduction, thank you for the invitation. And as it was just said, I'm interested in trying to understand the relationship between genotypes and phenotypes with a focus on protein post-translation modifications. And I'm going to explain this in more detail. But just to give you a general sense of what I'm interested in, you all probably know, I understand. I imagine that most of you come from biology, but if you don't, you can also then at some point ask me more questions. But as you all know, cells can do quite a number of different functions, such as the cell cycle or trying to find a microorganism in chemotaxis. So I'm very interested in understanding how does the process of evolution, so how you can have random mutations leading to the discovery evolutionary terms, how do these cell functions arise? And then how do they change across individuals of the same species? So how are we different among the same individuals of the same species? Or how do these processes change in disease? So you have a mutation that causes a disease. How do we understand mechanistically what's going on? So the group broadly tries to study this, the relationship between changes in DNA and changes in phenotypes. And then within this broad umbrella, more specifically, we also look at understanding post-translational regulatory systems. So I'm going to focus more now on the second part, and I'm not going to talk much about the broader aspects. And just to make sure everybody's on the same place, I'm going to briefly describe what we mean here by protein post-translational modification. So once a protein is produced, which is very simply schematized here with this diagram, it can still be modified by addition of post-translational modifications, such as the addition of a phosphate group, typically to serines, tyrines, or tyrosines, so the amino acids in those proteins. And this is catalyzed by kinases and can be reversed by phosphatases. And this is a way that the cells have to very quickly change the function of a protein. So the cell can sense something from the outside of the cell, and there may be a receptor, and then there can also be a cascade of information, such as one kinase can regulate other kinases. And this is already a process that allows the cell to make a decision to decide to whether change something inside the cell or not. And then this will lead to changes in protein activities and functions. So this has been studied since the 1960s. There's so many decades of study that since this process has been discovered. What has changed in the last 5 or 10 years is that now you can use mass spectrometry, which is a fairly recent technique to use this to study protein phosphorylation or ubiquitylation or other types of modifications. So now in one single experiment, you can identify and measure the changes in protein phosphorylation for thousands of phosphorylation sites in a single experiment. This is the thing that's new. And that's why now it's useful to use bioinformatics. So it used to be in the past that you would study one phosphorylation at a time. So now you study thousands, and therefore you need computational approaches to make sense of this. So there are many questions that one could ask of this data once you start having data sets like this. And we've been working on many different aspects of this. So I started looking into this by studying the evolution. So how conserve our protein phosphorylation sites across species? How does this change across evolution? And this led me then to study how important they are. And I'm going to talk about this. And then more recently to try to understand how these signaling systems work. So how complex they are, how do they change in certain conditions, and how do they change in disease? I'm going to give you a few examples of projects that we have done around these topics. So to start, I'm going to briefly summarize some of the findings that we have from evolution, which together with colleagues, we looked at phosphorylation sites across multiple species of the same species. And I'm just schematizing what we think is happening based on the data. I'm not going to go into the results, but what we think is happening is if we look across evolution, the phosphorylation sites are very rapidly changing across species. So it's very unlikely that the phosphorylation sites that exist in human are very conserved across, for example, mouse and further more distant species. So there's very rapid divergence of phosphorylation. However, the kinases, which are the enzymes that do the modifications, they are typically extremely conserved in their specificity. So a kinase often recognizes a few amino acids surrounding the phosphorylation site, and these preferences are often extremely conserved. And also the same kinase will typically do the same function. So a cell cycle related kinase is typically also going to be a cell cycle related kinase in another species. But the actual targets that kinase is phosphorylating are very potentially quite different across the species. So that's the observation. So then one question that comes from that is, what does that mean in terms of function? So are the sites changing and retaining function, or are there some sites that simply don't have any function at all, and therefore it doesn't matter if they change because maybe they're just simply not doing anything? So it's quite possible that there are many of these modification sites that are biochemically real. So if you do the experiment twice, you'll see them. So you'll find them phosphorylated. But in fact, if you were to mutate them or something like this, it wouldn't have any impact. And so I think that there is a substantial amount of protein phosphorylation that doesn't do anything. And similarly to the idea that there's probably also many splicing isoforms that don't do much, there's much in biology that is not selected for. It just exists because of the diversity that is generated throughout evolution. And so my group has been working on a number of projects to try to figure out which of the many phosphorylation sites we think are most important for the cell. And to give you some ideas of the problem, so for example, in yeast, there's around 10 to 20,000 phosphosites that have been discovered experimentally through mass spectrometry. And for human cells, there's an order of about 100 to 200,000 phosphosites that have been discovered. And then if you go through the literature, and there's databases that accumulate this information, less than 5% of these phosphosites have a known function described in the literature or have an annotated kinase or phosphatase. So it doesn't mean that 95% of these phosphosites are not important, but we just don't know which ones are most important. And so how do we start to study 100,000 phosphosites? So here are some examples of projects where we've been trying to address this question. So one way, and quite the simplest way, is simply look at phosphosites that are conserved across hundreds of millions of years. So if they're that conserved, they're very likely to be important. So we've done this in a project with Martha, a student in the lab, and this works really well, but unfortunately, there's only about 5% of phosphosites that are this conserved. So although this is quite informative, it doesn't cover a lot of phosphosites. Another approach that I'm not going to talk to you about is we also do a small amount of experiments in the group. About 1/3 of the group is experimental. And so one thing that we've been doing is generating libraries of mutants in yeast. So we make mutants, hundreds of mutants, where you mutate one phosphosite at a time and study how that impacts on fitness. And that also gives you another indication of which ones are important. But of course, this is experimental. It's a bit does more work to do. One approach that has worked very well is using computational methods, machine learning in particular, to try to rank which phosphosites we think are most important. So I'm going to describe this now. So if you have done machine learning, you may be familiar with the sort of things that you may need to do in order to do this. So in order to do this, what we needed was to find, first of all, a set of reliable human phosphosites that we know for sure are really true. I'm going to explain how we did this. And then we need to train a predictor. That means out of these 100,000 or 120,000 phosphosites, we took from the literature which of these have an annotated function. There's about 2,600. I think the number here is wrong. And then we need some sort of feature that helps us say which ones we think are most critical based on this training set of about 2,000 that we know for sure are important. And the features that we've derived are all based off just things that we know from previous years of work. So for example, one obvious thing is that if the phosphosite is extremely conserved, as I just said previously, then it's more likely to be important. We also have compilations of previously published experiments where somebody's done an experiment and measured how the phosphosites are changing in some condition. And so this allows us to know, for example, if a phosphosite is very often regulated across many perturbations, which again would likely indicate that this is an important phosphosite. We also have quite information a lot about protein structure, accessibility across the human proteome. So this also may be informative in saying if this phosphosite is very critical or not. So I'm going to show you how we put this information together. But first of all, one thing that we did to find, we think, are phosphosites that are very real, so biochemically well-characterized phosphosites, which may or may not have functions, was to reprocess data sets that were already deposited in the PRIDE database, which also is at EBI. And why is this important? So if you've studied phosphorylation, you may be familiar with a website called Phosphosite Plus. And what that database does is every time somebody publishes an experiment, they collect the identified phosphosites and they add it into the website. And the problem is every time somebody does an experiment, you have on the order of something like 1-2% false discovery rate. So there's about some amount of error in each experiment. And if you simply accumulate all the experiments without reanalyzing them, you accumulate the error because all the errors are going to be different. Everything that's real will start to be similar across the experiments. So the error rate as you accumulate experiments is going to be higher and higher. So that's why it's important then once there's many experiments, it's actually quite important to reprocess the data altogether from scratch from the raw mass spectrometry data to identify the phosphosites. And so this is what we did. And if anybody here has done mass spectrometry, this was actually quite harder than it sounds like, because just the amount of information that was used. So in total, this corresponds to something like 1 year of continuous mass spectrometry time. Of course, this was not our work. This is all work from the public domain that was available in the PRIDE database. And once we ran these analysis, we identified about 100,000 unique phosphosites that we're very confident in that these are real human phosphosites. Another thing that was important for us is that we had many sources of data. So there were many different human cell lines or tissues in the compilation. And most often when people do these experiments, they often would use HeLa cells, which is a very common cell line that's used for these studies. So what we wanted to know is if we were to remove these very commonly used cell lines, would we still get back most of the phosphosites? And this will tell us that we're not extremely biased by these types of cell lines. And that's what's shown here with the gray line, the rest. So if we take out these most commonly used cell lines, on this y-axis is the accumulated number of phosphosites. So we get more or less the same amount of phosphosites, even when we remove these very commonly used cell lines. So then we have this list of highly curated list of human phosphosites. And we added in these features that we've collected in the lab for over the years. This is just, this is real example, but it's not a very common example. Here, I'm showing the region of a protein called MAF1. And at the top, we have 1, 2, 3, 5 phosphosites. And marked in red are phosphosites that have a known function described in the literature. The other 3 sites may or may not be very important. We don't know that. And so what we can see is that in this particular example, these 2 sites that have a known function are more conserved across species. They are more often regulated, which is shown here in this plot. And they have more mass spectrometry evidence that they are seen in multiple tissues or just simply more counts, more times. So this is this is just an easy example to show how you could take these data and integrate it using a machine learning predictor that accumulates the evidence and says, yes, if it's conserved, regulated across many tissues, and so on, is more likely to be functionally important. That's essentially what we did. And one thing that while doing this, what we see as well is that there is no single feature that can be very predicted by itself, which is shown here in these plots. So the blue distribution is the distribution of scores, in this case, for example, for conservation, for phosphosites of known function in blue, or all other phosphosites in red. And what we'd hope is that there's some features or combinations of features that can discriminate well the phosphosites of known function from the background. And there is no individual feature alone, but then we can combine them so they're very orthogonal. So we can actually combine them in a very useful way. And this is what we've shown here. So in the paper, we describe different attempts. We use random forest, different linear regression models, and so on. So the actual method of integrating here doesn't make a huge impact on the outcome because there are not many features and we have a large training set. So it doesn't seem that there's one machine learning approach itself that outperforms strongly the other ones. But the important thing is once we combine all these scores together, we get what we call here the phosphosite functional score, which is shown on this axis. And on the top in blue, you have the distribution of scores for phosphorylation sites for all phosphorylation sites. So these 120, more or less. And then in these orange colors, we have phosphosites that have a described function in the literature. So we get a pretty good discrimination between these 2 distributions. So, and we also can break this down into different types of functions. So even if you have phosphosites that regulate enzymatic activity or localization or other properties of the protein, we get reasonably good discrimination, which is good because it doesn't mean that we are sort of very specifically focused on one type of function. What this is bad is that we cannot really tell you. We can say that a phosphosite is very important, but we cannot say that a phosphosite is very important because it's doing something specific to the protein. So the other thing that we looked at is phosphosites that are in positions in the protein, then when mutated cause a human disease, which is these 300 shown here in green. And so this is information that was not given to the model. So these are phosphosites that are in positions that cause human diseases when you mutate them. And these are also scoring even higher than these phosphosites of known function. Again, this is not information that was given to the model. So it's a good way of showing that we can use this to rank critically important phosphosites. We wanted to have yet another orthogonal set because, in a sense, this data set was used for training purposes, the ones in orange. So we tried to find another different data set of phosphosites of known function, which is, we took advantage of information that exists in UniProt. So we looked through UniProt for cases where somebody has done an experiment, so somebody has done a mutation experiment, and they recorded either if the mutation causes some effect or no effect to the outcome. And the outcome could be a function of the protein or a function of the cell. So then we take these about 1,000 phosphosites that were mutated in different experiments, we bin them across this functional score. And here I'm just reporting the ratio of effect over no effect, either for a loss of function in orange or a gain of function effect as reported in the literature. And so what you can see is that the higher this functional score, in particular, say above 0.5, 0.6, you see a very strong enrichment for cases where a mutation is reported to cause some effect of the protein. And then there's some interesting differences between the fact that some alanine mutations are mostly reporting loss of function and negative charge mutation, which mimics a phosphosite, also is reported to cause a gain of function, and we can also equally predict based off the functional score. So these are all, in a sense, taking advantage of public data to show that the method works. Of course, many times if you want to impress that this really works, you ideally do some new experiments. We don't do human experiments, but we happen to have many collaborators that are very good at this. And I'm not going to show you the details of this, but then what we try to do is ask another group that also works at the EMBL, so Min Nose Lab, and they are interested in neurodegeneration. And so we looked at one protein of a complex that they're interested in, which is the SWISNF chromatin remodeling complex. And this protein, SMARCA-C2, is expressed during the development of neurons, and then forms part of this complex during neural differentiation. And this protein is heavily phosphorylated, as shown here in this diagram. Each of these lollipops is a phosphosite. And the higher that is, the higher the functional score as we predicted. So then we took the two of the most highly scoring phosphosites that were near each other, and Askar and Tony in her lab, they generated alanine mutations, so homozygous double alanine mutations for this protein. And then they took these homozygous, heterozygous, and controls, differentiated them to neuronal lineages, and through gene expression in microscopy, they just checked, are these neurons developing properly or not? And from both types of experiments, we could see that there was a delay in neurodifferentiation when these phosphosites are mutated to a alanine mutation. So it really indicates that these phosphorylation sites are important for the function of the protein in the context of neurodifferentiation. Okay, so what this is a summary of the project. We reprocessed a large amount of mass spectrometry data to generate a very comprehensive list of human phosphosites. If you're interested in phosphorylation, there is, as I said, this database called Phosphosite Plus. And I think now it's starting to be an issue that they've accumulated so many of these data sets without reprocessing the data that I think there's an increase in the false discovery rates in that compiled data set. So there's likely to be a reasonable number of errors in that website now. So then we integrated different features that tries to come up with a single score that tells us which of these human phosphosites is most important for the cell. And we think this really works. So if you take the top of the list, and these are more likely to be important, but we cannot say what is doing to the protein. So we cannot say the phosphosite is regulating, for example, an interaction or an activity or localization. There are some of the features in the data that we are using that is somewhat informative. So for example, we use protein interfaces as one of the features in the model. So if a phosphosite is scoring very highly and it's an interface between two proteins, then of course that's already telling you that perhaps the function of it is to regulate the protein interaction. The other thing that the model doesn't tell you is if this phosphosite is regulating DNA damage or cell cycle. So what is the biological process that it's regulating? And we tried So we tried some models to do this, but they work okay, but not particularly well. So I think there's an area there to try to explore. And so that tells you how we try to figure out which of these phosphosites is important. So now I'm going to show you some work on trying to look at how these phosphorylation sites change in disease, and how can we use this information maybe to say something about drugs that could be useful for treatment purposes. Then I'm going to start with a very topical area, which is looking at how phosphorylation changes during infection of SARS-CoV-2, which of course is something that has been on the mind of many people. And this is a very interesting project for many things. And I should say from the very beginning that this is a very large collaboration effort involving many different people. My group was involved in really the computational aspects of the work and helping out the coordination. And it was a very exciting project, I should say. We, from the start of the experiment to the manuscript being published, I think it was something like 3 months, which I've never done this in my life. It was really just the speed at which we had an experiment done, we analyzed the data, we proposed a new experiment, and then the week after we had results, and sort of the pace that this moved was really exciting. So this was spearheaded by Mehdi, which is a postdoc in Evan Crooken's lab. And as I said, there are many different groups involved in the work. And the idea here is straightforward. So the idea is, if we understand how the cell is changing during infection, can we suggest a drug that would counteract that effect? And does that help to stop the infection? So what was the experiment then? So the experiment was we compared cells that were infected with SARS-CoV-2 or with mock. These were Vero-6 monkey cells. And then these were incubated with the virus. And at different time points, samples were collected from 0 to 24 hours. And then these were used for mass spectrometry, either to obtain protein abundance measurements or either to take protein phosphorylation changes over time. So one of the observations that I'm not showing you here is that there's very little changes in protein abundance levels. So there's the most striking increase in protein abundance is the viral proteins that are very produced starting about 4 hours. But this is known that the virus stops the production of host proteins in order to favor the production of the viral proteins. So most changes that we see at the phosphorylation level are not explained by changes at the protein level, because we don't see protein levels changing. So then what we wanted to do is we can take these phosphorylation sites and we wanted to estimate which are the kinases that are being regulated. And without explaining you technically necessarily how we do this, this is described in these previous publications from our group. Essentially, the idea is if we take the targets of a kinase and they're all going in the same direction. So if all the targets of a given kinase are all going up during infection, this suggests that they are becoming increasingly activated. The kinase is becoming increasingly activated. And then technically, there's many statistical ways that one can use. In fact, this is described in this bioinformatics paper. The most boring of ways is actually the most one of the most accurate, which is really just taking the median change of the targets and using a z-score. We tried much more complicated models, but that in fact works pretty well. So if you're interested in those technical details, it's described in these papers. But then the outcome, the biological outcome of this work is we could predict the activity changes of about 100 human kinases during this process. And what we see is that there's a increased activation of casein kinase 2. There's increased activation of kinases belonging to the p38 pathway, downregulation of several cell cycle kinases, and downregulation of growth-related signaling kinases and some cytoskeleton-related kinases. And so there were two things that we wanted to do with this. So on one hand, can we use this to understand the biology of what's happening inside the cell, and then use this also to predict drugs that could counteract these changes. So one of the things that we could see from the lab experiments was, as I said, that there were several changes in regulation of phosphorylation sites that could be related to cytoskeleton reorganization. And we teamed up with a group in Freiburg, Robert Koster's lab. I knew that they were working already with the infected cells, so I told them that we had this evidence from mass spectrometry data that suggested there were several kinases that could be involved in regulation of cytoskeleton. So we asked them to have a look in the microscope to see if there were aspects of cytoskeleton that were changing during infection. And so in fact, this is what they've shown then in these infected cells, there's increased production of these phyllopodia structures. So these are sort of fingers that are protruding away from the cells when they are infected. And these at these protrusions, you find the protein, in this case, M protein from the virus, suggests that this may play a role in egress, so how the virus escapes from the cell. So based on some previous experiments from a previous project, we also knew that the N protein was interacting with casein kinase. So N protein from the virus was physically pulling down the casein kinase, which we thought could be involved in the regulation of these phyllopodia structures. So what the Roberts lab has done was to then try to localize in these protrusions. He checked whether you can find casein kinase 2 and this N protein, and in fact he did see this. So at the tip of these protrusions, there was co-localization of N protein and this casein kinase 2. So the other thing that we've done in the lab is just overexpress N protein by itself. And that was sufficient to cause an increase in phosphorylation of casein kinase 2 targets. So the current hypothesis is that via the N protein of the virus, there is an increased activation of casein kinase 2, which leads to the production of these finger-like structures in the cell. And this is as far as the project went, so we didn't really have time to explore this more, but there are many questions. So one of the questions is, in fact, is this hypothesis true? So if one were to inhibit casein kinase, would these structures disappear? So we didn't really test that. And the other question is, what does the virus need this for? So some of the ideas is that this could be useful to infect nearby cells, or simply to increase the rate of budding so that it offers more surface area for the virus to escape the cell. So I don't know why I have this. So the other piece of data that I told you about is that we saw this decrease in kinase activities related to cell cycle. And I'm going to briefly explain another way that we have to analyze phosphoproteomics data sets. So I mentioned in the past already that we have many data sets compiled from previous studies where somebody has done a perturbation experiment. So for example, somebody has done an experiment where they treated cells with DNA damage, and they measured how phosphorylation sites change in response to DNA damage. So we have many hundreds of these experiments that we've compiled in the lab in the past. So if any new experiment that comes in, such as this experiment with a virus, we can compare how does the phosphorylation changes induced by the virus infection look like compared to, say, DNA damage or any other condition that exists in our compilation. So when we did this, we could show that that infected cells have a change in phosphorylation that is very similar, so very positively correlated here in red, with the cell cycle stage that's the S to G2 transition, and it's the complete opposite of a cell arrested in mitosis. So it looks very different from a mitotic cell, and it looks very similar to a cell that's been arrested in S and G2. So this suggests that the virus is somehow arresting cell in a particular cell cycle stage. That was then an hypothesis that we could test in the lab. So then this was tested by infecting cells and then just measuring with FACS DNA content. So using this, you can very simply see how many cells are in different stages of the cell cycle. So comparing mock with infected cells, we could see that there was an increased proportion of cells that were either in S or G2-like states arrested before mitosis. Which, again, this just tells us that the virus is trying to stop the cell cycle, but we don't know necessarily why is it doing that. There are other viruses that do this. HIV also does the same thing. And there's some discussions in the literature that may have to do with availability of nutrients and things like this, but we don't really know necessarily why this is happening. And so finally, one aspect that I was very excited about in the project, maybe not others in the project, but something that I find interesting, was that we saw that the virus induces the activation of the PTMs pathway. Why could this be interesting? The downstream of this PTMs pathway, there's the production of interleukins. And so one of the aspects of the disease, so very severe disease of COVID-19, is that there's a very hyperactivated immune system. And so the hyperproduction of BL6 and other interleukins has been associated with a very poor prognosis. So people that end up being very sick sometimes are really having a highly stimulated immune system. And so we thought this could be one of the potential reasons of why this may happen. So by some unknown mechanism, there's an activation in p38 leading to the overproduction of BL6 and then an overstimulated immune system. So Jeff Johnson at Mount Sinai confirmed that through other approaches, so using Western blots and antibodies, confirmed that in fact there's an hyperactivation of the p38 pathway during SARS-CoV-2 infection. And then measuring the production of BL6 here by looking at the RNA of BL6, you could show that in fact there's an increased production of BL6 for when you infect cells with a virus. But then if you at the same time inhibit p38 with increasing concentrations of the p38 inhibitor, you see that there is a decrease of production of BL6. So this increased production of BL6 by the virus depends on p38. And I'm going to show you just at the end. In fact, if you inhibit this pathway, you also end up stopping the proliferation of the virus. So then this is a summary of just the work. So what we took from this is that we could see which kinases were very often regulated during infection by SARS-CoV-2, which then allows us to match kinase inhibitors. So based on those kinases that are most strongly regulated, Kevin Shokat at UCSF just picked out which kinase inhibitors would be most appropriate to use. And of course, we don't know a priori if we're going to inhibit that kinase. It may be that it actually makes the cell even more susceptible to the infection, or maybe it stops the infection. So we don't know if that change in activity is important for the virus to proliferate, or maybe it could actually be something that the cell is trying to do to counteract the viral infection. But at least it gives us a list of kinase inhibitors to try. The other thing that we did is we used this functional score that I described just previously in the other project to look at phosphosites that we thought could be highly important. And so this allows us to look for regulation of other enzymes, so not protein kinases, but maybe other enzymes. And so, for example, we found a phospholipid kinase that seems to be strongly regulated based on this approach, which again is another way that we can use to find drugs that could be used. And so based off of this, we found something like 87 drugs that we could test. And without showing you much of details at all, you can look it up in the paper. But then Marco Vinuzzi's lab at Pasteur Institute and Adolfo Castillo-Sarte's lab at Mount Sinai, they've been doing a lot of these experiments where you take a cell line that's been infected with the virus, you treat it with the drug and see if that has an impact on proliferation. So there are many of these 87 compounds that we suggested. There are many that actually inhibit the viral proliferation. I'm just going to showing you here the ones that relate to the p38 pathway, because that's one that I'm interested in. And in one of these, gilteritinib or other AXL inhibitors are now in clinical trials for COVID patient disease. So yeah, so this is just a way to accelerate the identification of compounds. And the idea, of course, if we have a drug that's already being used for some other application, then we know it's already safe to try to use it in humans. So that's also the rationale that we try to use. We try to find drugs that could stop the infection in the cell lines and already being used in the clinical trials for something else. So then this is easier to propose for clinical trials for COVID as well. So this is one of the projects, I think. So in total, we were involved in about 3 of these projects. And out of 3 of these projects, there's now 26 drugs that we've shown in cell lines to be effective against COVID and are now in clinical trials. So not all of these 26 are in clinical trials because we suggested them. Some are already in clinical trials, but some of these were moved to clinical trials because we've shown some evidence in the lab that they work. This made it to the news, and unfortunately, the thing that was picked up the most was this idea that the cells were having some sinister tentacles. So not really the message that I was most excited about, but I think this is just the thing that was most in the mind of people. Okay, so just in the last few minutes before I as you ask questions, I'm going to move to a project that's unpublished. And it's similar in the fact that we are trying to find how phosphorylation changes during disease, but it's different in the way that we try to do that. So if you're interested in bioinformatics, cancer data sets in general are very interesting because there's a lot of publicly available data. So you can do a lot of analysis without having to do experiments, new experiments. You just, if you have a good idea, there's a lot of published data already available that can be reused for your purpose. And so if you're not already familiar, maybe something that you should be familiar with. So for cancer data sets, you have matched data. So for the same samples, more or less, you have information that comes from either the genome, such as mutations or copy number changes. You have RNA levels or methylation levels. You have now also another project where for many of the same samples, you have full proteomics data sets, so protein abundance and phosphorylation data. And for a larger number of samples, you have a more limited type of data, which is antibody-derived protein abundance or phosphoproteomics data. And then alongside the tumor samples, we have also a lot of cancer cell lines. So these are cell lines that are cancer-like, so they can be potentially matched to a tumor. And for these cell lines, there's also quite a lot of sort of genomic, gene expression, protein, and increasingly proteomics data, and also a lot of genetic data, so CRISPR or shRNA or drugs. So you can use these data sets for many things, and we've been playing around with these data, and so many people around the world play with these data. And it's very interesting, of course, to study cancer, but also to study biology, because these are extremely messed up cells. And so the concept is that you can take advantage of the fact these are very messed up cells to figure out how the normal cells work by taking advantage of this. So you can make many statistical analysis by taking advantage of the large number of samples as well. And so one idea that if you're not used to thinking about it from the genetics perspective is because we have also the DNA and how the DNA is changing, this allows us to say something that's causally linked in terms of the associations. So let me explain this. I think this is a useful concept if you're learning about bioinformatics. So oftentimes when you do some correlation analysis, so an obvious thing would be that you, for the same gene, you could correlate the RNA levels of the protein of the gene with the protein levels of that same protein. Or maybe the RNA levels of one protein against the protein levels of some other protein, for example. So if you find a correlation like this, you don't know if what's causal here. So you just know that there's some association. It could be that in fact the RNA levels of one thing cause a change in the protein levels of another thing. It could also be vice versa, or it could be actually some other thing. So some other change that causes both of these, and then that's why they're correlating. So that's why oftentimes people will tell you correlation is not causation. And of course, that's a very important thing to keep in mind, because oftentimes you can have spurious correlations that don't tell you much about the causality of it. However, if you're correlating a change in the DNA against something like a molecular change, such as the protein levels or the RNA levels, then that correlation is much more likely to be causal. And that's simple intuition about how biology works. So if you have a mutation and you see a change in the protein levels, it's biologically very hard to think of a mechanism by which the change in the protein levels cause the mutation. It's much more likely that the mutation causes a change in the protein levels. So by correlating things with DNA changes, you add in this causality aspect to it, which is potentially very useful. Right, so in these data sets, one thing that we often use is the copy number levels. So in these tumor samples, a gene in the DNA may actually have been, for different reasons, occurring in multiple copy numbers. One way is that the whole chromosome can duplicate. So then you have, instead of having maybe 2, you have 4 copies of that gene. Or it may be that you have a local amplification or a local deletion. And so a gene can occur in multiple copies in the DNA. So then you can use this to make correlations, as I said, that are causal. So you can causally associate something with something else. The problem is that these amplifications or deletions don't occur as a single gene. So most often they occur as a large block or even a whole chromosome. So then it becomes tricky to say what is it in that region of DNA that was duplicated that causes it. So you have to come up with a way of doing that. And as any other genetic association, so not just this, but any sort of genetic association, these are very often fuzzy, meaning that there are potentially many steps between the change in the copy number of a gene and, say, the correlated change in the protein level. So there are many steps in between these things. So although it's causal, it doesn't mean that it's direct. Okay, so then I'm just going to briefly, just in a couple of minutes, tell you what the project is about. It's something that's in press, going to come out soon, I hope. And the idea is, can we find copy number changes in the DNA that are causally linked to a change in the protein phosphorylation states of a protein? So can we find a gene that we know for sure is causally changing the phosphorylation states of some other protein? There's about 4,000 samples. The disadvantage is, because we're using the antibody data, there's really only a few phosphosites that we can look at. But then we can look at the whole genome affects those phosphosites. So the phosphosites that we can look at is all the ones that are available in the RPPA. Again, this is all publicly available data. Everything that I'm going to tell you about here. If you're interested in signaling, which I'm not sure how many of you would be, these are the phosphosites and the signaling pathways that we could look at. So in technical terms, how one does this, so technically this is, we look at linear models and we do a log ratio test comparing two models. So we want to know if a copy number change in gene X is affecting the phosphosite levels of protein Y when we take into account the protein levels itself. So if a phosphosite is going up, it could also be just that the protein itself is going up in abundance. What we want to know is that the phosphosite levels are going up, taking into account already that the protein levels are maybe up or down. And we also take into account other covariates such as the gender, age, or tumor type, just to make sure that we're not doing something that is indirectly associated with these things. So once we do these tests, essentially we have this problem that I tell you about, where this is one example. So this is one phosphosite in particular, and then across this axis we have the whole genome. So different parts of the chromosomes from one point to the other. And as I said, what happens is that you have copy number alterations that are whole regions of the chromosome. So here's around chromosome 3, for example. You have, so each of these points is one gene along the chromosomes, and then this is the strength of association. So there's a group of genes here that are all close together, and the reason it being that, maybe there's one of these genes that's causally linked to a change of phosphorylation sites, but then there are nearby genes that are co-amplified and co-deleted. So you don't know exactly which of these ones is the actual causal one. But there's one of these that's likely to be causal. So then what we needed to do is pick one of the genes in each of the regions. So within each of these regions, we need to pick one that was the most likely one to be causal. And there we did a couple of different things. The simplest thing is just we could pick just the strongest signal within that region. And that was often the easiest and most obvious thing. So PTEN is a phosphatase that is known to regulate the phosphorylation levels of AKT at this phosphosite. And this is really the strongest signal that we see in the data. But then in the other regions, there maybe was not as clear. And we added in a couple of other features. Another feature was similar, looking at correlations also by gene expression. So not just copy number, but also gene expression, just to add support. And then we also look at the signaling network, and we look for genes that were closest to that phosphosite in the signaling network. So we took a signaling network and we just count the shortest path between each gene and the phosphosite harboring protein. So when we put these different together, we can come up with a single score that can do this. And we try to make sure that the associations that we find are reproduced across different tumor types. And also, because there's data on cell lines, we can also try to reproduce the association in cell lines. So we end up with a fairly short list of about 200 in total, and then out of these we can be more certain about, say, 40 of these, more or less. So it's a fairly short list, but then we make sure that it's reproducible in multiple tumor types and in cell lines. And out of these, there are many that are previously known, like the example of PTEN and AKT that I just showed you about, but there are also potentially other ones that could be interesting. And so besides learning about biology, so one thing that we learned about the biology is that, you know, for example, as I just said, so P10 changes in protein in expression levels of P10 are linked causally to changes in phosphorylation of AKT. So we learned some of these things. The other thing that we hope to learn is there maybe some things that are worth using as drugs in specific contexts? And so the way we looked at this was to try to find cases where the cells get addicted to high levels of kinase activity. So why is it that in some tumors we have high levels of phosphorylation of some proteins? Oftentimes it's because that the tumor is needing that activity in order to become a tumor. And one way that we have to study this is the cell line data that I told you about. So for many human cell lines now that are tumor-like cell lines, we have measurements of phosphorylation, the same measurements from the antibodies. And we also have measurements where every single gene has been knocked out with CRISPR or shRNA, and measured whether the cell lines die if you lack that gene. And there's also many evidences from drugs. So if you treat the cell with a drug, whether the cell dies or not. So then there are more or less sophisticated ways of analyzing this. A simple thing would be simply to correlate the phosphorylation levels of the kinase with the survival of the cells when you inhibit a drug or when you try to knock out a gene. So based off of this, we can find evidence, which is shown here, that if you correlate the activity of a kinase with a knocking out or knocking down that kinase, you typically find that when the phosphosite tends to activate the kinase, there's a positive correlation, and when the phosphosite tends to inhibit the kinase, you see a negative correlation. So it does seem that there are many cases where the cell becomes addicted to that activity, and this happens for some kinases more than other, or some genes more than other, which is shown here. So in particular for AKT, YAP is a transcription factor, MK is a kinase, CHEK2 is a kinase, and so on. So there's a couple of kinases and transcription factors whose activity is important for these tumor cell lines. So if you drug them in the right context, then in principle you should be able to find drugs that are good at stopping these particular cancers from surviving. So this is shown here. So then if you put these two ideas together, what we can do is we can find biomarkers, which is shown here in this plot. So from the first thing that I showed you about, we can predict whether, for example, the expression of RBB2 is associated with increased activity of EGFR receptor. And then if this activity is important for the survival of the cells based off knockouts or drugs. So by putting these things together, we can find a biomarker, in this case, RBB2 expression, that's a biomarker for the activity levels of a kinase that then suggests that you can use specific drugs to kill these tumors. So that's the general idea of this project. And because it's already pretty late, so then in the paper, we tried some few new things that were not already known from the literature. I don't think this is yet something that now could go to the clinic. For example, I'm not suggesting that we have a very powerful way of finding new drug candidates for treatment of cancer. So I think it's more of a way of figuring out new associations between copy numbers or particular genes and the activity of some kinases. So I'll skip this and just give you the summary. So we find that there is a way that we can find copy number associations with kinase signaling. So we find these novel associations. We can potentially use either these copy numbers or phosphosites as biomarkers. And now we're trying to do this now with not the antibody-based data, but with the larger scale proteomics, phosphoproteomics data sets that are available. And with that, I will stop. Thank the collaborations, the funding, group members, and I'm happy to take questions in English or in Portuguese.
Thank you, Pedro, for your inspiring presentations. So I'm gonna open to the audience to questions. Please send the questions on the chat or speak through the microphone in English or in Portuguese, please.
Pedro, thanks for the talk. And I was just wondering, the first thing you presented was this functional score that you developed. and I found it very interesting. But I was wondering about the false negatives you find. How did you work with that, and did you have many? What is the number of false negatives? Because I was thinking about that result where you picked the highest scoring functional proteins, and you checked if it mattered on neural development. And I was looking at the little ones and thinking, well, are they actually negatives, or could they be something else?
Yeah, it's a good question. And this whole problem of trying to say that a phosphosite is important or not important is very difficult, because we can never prove that something does not have a function. So we can never prove a negative. That doesn't exist.
But did you think or consider looking at picking random, low scoring ones just to check? I was just thinking, could they be important to you? Do they add up to this change in neurodevelopment or whatever?
Yeah, these are all good questions, and it's fortunately not very easy. So one thing that we did in the yeast case, where we can do the experiments, is we mutated 500 phosphosites, so 1 of 500, one at a time, and we tested 100 different growth conditions. And again, we can never say that if we do 200 growth conditions, we're going to get more. So we don't know that. So, but for these 100 different stresses, there's about 40% of phosphosites that have at least one growth defect in at least one condition. So it's very clearly going to be the case that some are more important than others, and it's very hard to actually say what's the false negative rate, because we don't have a real negative. So it's extremely very difficult. So all that we can say is that we have something that we can think ranks them by order of importance. And if you want to do an experiment, you're better start at the beginning. So leave the end of the list for last. That's the only thing that we can say.
Okay, thanks.
Thank you for your question. Well, I had one question, Pedro. I was curious. The deregulation of protein phosphorylation is more correlated to mutations at the actual phosphorylation sites or the nearby sites on the protein? I mean, concerning the mutation frequency, is this later information already know or can be assessed or predicted somehow?
So we don't know. So we can definitely make predictions. So for example, we can use this functional score as a variant effect predictor. So if you're interested in disease mutations, there are many tools such as SIFT or Polyphen or other things like this. So we can compare if we just look at the actual phosphosite position, when we compare our functional score with other tools, our score there is better when we try to predict disease mutations. We can also look at the allele frequency in the human population. So if it's a very rare allele or it's a very common allele, then a very common allele in the population should not hit a phosphosite that's very important. And this is true in the data. We didn't look in the vicinity, for example. So we didn't look much whether we see signs for constraint around the phosphosites that are more important, for example. But it could, I imagine you would see that, but we didn't look at that.
Yeah, I was trying to look. I was thinking, try to look that through the protein structure using PDB structures that are already known for the phosphorylation sites. I'm thinking something, but I don't know if it will work about that.
Yeah, so we compile homology models and structures, and we place the phosphosites in the structures, and we have different studies around that. So in fact, one study that we're trying to do now is more, we would like to be able to say when a phosphosite changes the structure, which is something that there's very little data for this. If somebody's interested in a problem, this is a very interesting problem. A very hard problem as well. But.
Thank you. So we have another question?
Yeah, I would like to know, Pedro, you have a lot of data that people would like to use bioinformatically. Do you keep a website or a report of all the data data that you have, scores for phosphorylation, etc. Do you deposit it anywhere?
So we always try to make the data available in some way. So for example, for this functional score, in the we don't have a website, but in the supplementary tables of the paper, you will find all the features that we use for training. So you could train the model. Actually, we have also in GitHub, we have a tool that you can change. So we made a small tool that you can use to train your own predictor. And of course, you can go in and play with that predictor and change it. And so the features are all there if you want to change them.
No, I know. That's great. But I would trust the application and would like to assess the list of proteins, the score, etc.
Yeah. So
you have this as a supplementary material, right?
Yeah. So that's all as a supplementary. Yeah. And so for other projects, for this collection of phosphosites that are changing in different conditions, we also have a website. Because if you wanted to compare your new experiment with the previous experiment, you can put it there in this website and see which kinases are being regulated and how your condition compares to the other conditions.
Interesting. This is a difficult approach. I know that P53, for instance, when it gets phosphorylated, then it attracts P300 to bind and acetylate it. Do you see in the horizon the possibility of working at the same as well as you work with phosphorylation to have this work with acetylation?
Yeah, so there are 300 or 400 different types of PTMs. We could clone my research group like 2 or 3 times to work on this. Yeah, but if I think there's important challenges like this in other PTMs. I completely agree. And as you mentioned, there's this crosstalk idea. So oftentimes one PTM causes or inhibits another PTM. If somebody's interested, ubiquitin and acetylation would be the 2 ones that are worth recommend working on. So we work a little bit on ubiquitin, but mostly on phosphorylation. But again, if somebody's interested in sort of pursuing this area of research, phosphorylation, ubiquitylation, and acetylation would be the 3 ones that I'd suggest to work on.
Okay. And do you have some type of annotation of if a kinase or either a phosphorylation site is more promiscuous or is more specific? How can
I didn't go too much into this, but so one of the big problems, as I said, is figuring out which phosphosites are functionally relevant. Another very, very big open problem is figuring out which kinase and which phosphatase regulates each of those 120,000 phosphosites.
I know.
And so that's a huge, difficult problem. And we tried a few things. We have 1 paper published, but it's actually a very difficult problem. So it's an area of research that needs a lot of work. And the same is true for any other PTM. So ubiquitin and acetylation also have enzymes that make the modification. And again, there's basically nothing known about that.
Okay, so you cannot tell the specificity of phosphorylation site if it's too complex, it would need a special binding with the phosphate, or like protein kinase A and C.
Yeah, so we can make a ranking of which kinases regulate which phosphosites, but it's not a very good ranking.
Okay, you would not trust, yeah.
So I wouldn't make many experiments based off of that. So kinases like some things around the phosphosite. So some kinases like phosphosites, they have a proline next to it, and so there's all these rules, and you can encode them into a model. But even when you do the best models, I think they're still pretty bad. And even though I can report an AUC of 0.9 and things like this, but it's still pretty bad. And because the problem is that the space of negatives is much, much higher than the positives.
Any other question?
Yes, I'd like to make a question, please. So let me just open. So hello, everyone. Thanks, Dr. Beltrao, for your amazing talk. I was interested about something you stated about kinase activity changes in the cell may actually be good drug targets when someone is infected with COVID-19 or other viruses or something else. But it is still not known whether these changes makes the cell sicker or if it actually like a counterattack against the virus. So I was wondering if you have like any clue what approach could be done in bioinformatics to make it like more accurate against some drug possible targets, or if this phosphorylation can also be related to other situations like cancer or something like that.
So we tried this in a couple of contexts: HIV, salmonella, and oftentimes the kinase activity change is needed for the infection. So if we inhibit the kinase, more often than not, it helps to clear the infection or slow down the infection. But with salmonella, for example, we also had some examples where it was the opposite. So we actually inhibited and it actually made the infection worse. So it's just not easy to get the directionality right. It seems that if they are changing during infection, they're being important for something, but it's very hard to say if this is required for the virus or for the host. And I don't know of a good way yet of saying which direction it is. I'm guessing if we do this more times with different pathogens, we may start to see some ideas of why is it that some are required for the virus and some are required for the host. But it does work surprisingly well. So it works. So these changes during infection are better than the change that we see in tumors, for example. And I think the reason is that it's something to do with the fact that the virus is like an acute perturbation experiment, where you treat the cells with the virus, and over maybe minutes to hours, we see the phosphosites changing. And in the tumor samples, what we have is sort of tumors collected from patients that are in steady states. And biochemically, this is very different. And I think that's the reason why we have more success, I think, with infections.
Okay, thank you. So since it seems to be a little specific, it's hard to determine like a good drug test because there would have to be many drugs for many situations and many infections, right?
Well, I mean, I'm surprised by this COVID work. We actually found many inhibitors that had a good capacity to stop the infection in the cells. So, of course, then there's a whole process of figuring out which ones are going to work in a human, because you have the infection inside the host cell. It's a much more complex process. But it did work pretty well. The same for HIV. So we did find several drugs that were pretty good at stopping the cells, at least in a cell-based model. But I think working on the process of taking a drug from a cell-based assay to something that can be used in the hospital is a completely different thing. It's not something that we do or want to do.
Thank you again.
Thank you, everyone, for your questions. Any more questions?
I would like to.
Go.
Yes, Pedro, when you talk about in terms of kinase activity, are you referring to the FP binding? Because we have like pseudokinases that they still have some activity, although they are not able to transfer the phosphate to another phosphorylation, to another receiver. I don't know the word technically. So my question is about that. How the pseudokinases are considered as inactive in this way?
Yeah, so very specifically, what we do is we look at the substrates of non-substantive kinase. So I guess in the case of pseudokinase, I'm not sure that we would have any known substrates. And we just statistically check whether the targets of that kinase are all trending to be upregulated or downregulated in that condition. It's a very simple idea. So, if a kinase has 3 substrates and those 3 substrates are all going up in that condition compared to the background, then that suggests that that kinase is becoming activated. Of course, it's much more complicated than that because each phosphosite may have more than one known kinase regulating it. And so, in fact, we don't. It could also be that we're making errors because there may be phosphosites that are shared across kinases, And so we are predicting something incorrectly because we say that this kinase is up, but then in fact it should be another kinase that's going up. So it's a prediction, and as many predictions, there are some error rate associated with it. There would be another way. So just to be complete, another way would be to look at phosphosites in the kinase itself, because many kinases, in order to become active, they themselves need to be phosphorylated.
Yeah.
And so you could look just at the phosphosites in the kinase itself. The difficulty oftentimes is with mass spectrometry is we're only going to see. So I told you about 100,000 phosphosites, and in fact, in a typical experiment, we may see like 10,000, or if we're lucky, 20,000 phosphosites. And so we're probably not going to see all the phosphosites that we want to see. But if we have at least 5 targets of a kinase, that's probably already enough, even if we don't see all of the targets of a kinase. So that's just a way of helping us with the sparsity of the data.
Okay. Thank you.
Thank you for the question.
Thank you, Fernanda, for your question. Any other questions?
Oops. Fernanda is probably interested in kinases in plants. Do you think, Paulo, that it would be interesting to do the same kind of study in plants, even if in Arabidopsis thaliana? Because sometimes if you want to cure some illness in a human patient, you have to go like 100%. But with plants, if you affect the phenomenon in 5%, that will represent billions of dollars. So it does not have to cure the plant, but just modify. Do you know anybody that's doing the same thing with plants, or do you think on plants someday?
There's a lot of phosphorylation data for plants, actually, that Arabidopsis thaliana in particular, but there are a couple of other ones. We don't focus on that. We use the data from plants to say how conserved they are across species, but we don't really look at plant phosphorylation. One of the difficulties is that the relationship between the kinases and the substrates. I told you that 5% of the phosphosites have a known kinase in human. For plants, that's probably close to 0. So it's already difficult in humans, so some of these analyses would be very impossible to do in plants. But I agree that would be quite exciting to work on.
Someday. Okay, we are dealing with the reactome, and I don't know if we could just find all kinases in the website, but as we have the data in local MySQL database, we can try to investigate the kinases there, or the kinases substrates there. We are trying to look at the origin of genes, and I was inspired today of looking at the origin of kinases and also maybe the substrates. I have to learn how to assess the list of relevant kinases, right?
So kinases are very old, but so the evolutionary rates are very different. So kinases are very old and very conserved, but the substrates have evolved very, very rapidly, because it's the evolution of the targets are de novo. So you don't have to have gene duplication. So you have a new phosphosite, or you have a gain or loss of phosphosites without having to have a gain or loss of a gene.
Okay. I always thought that many functions are old, but if you go over them bioinformatically, sometimes you get surprised that some proteases are quite new, like in our saliva.
Yeah, so I'm sure that there are recent duplications and recent particular duplications events of kinase. I agree with you, there should be. I mean, I also interested. I mean, this is an interesting topic for evolution purposes, like how new genes arise and how they get integrated into the biological system.
Great.
Any other question? None on the chat. Yeah, we have one question on the chat.
Pode ler em português, está em português?
queria saber se há pesquisas, que é do Alexander Stacker, tá? Queria saber se há pesquisas que relacionam os eventos de fosforilação e as doenças relacionadas a um desarranjo e instabilidade da energia molecular e nos sistemas de interação interproteicos envolvidos. Talvez algo mais ligado à física em termos de pesquisa. Se uma fosforilação iniciaria uma cascata bioquímica em razão de uma instabilidade cinética.
Não sei se eu percebo completamente a pergunta. There is very little evidence of relationship between impact on phosphorylation and disease in general. There's not much known. But this is more complicated, right? Instabilidade na energia molecular do sistema de interação. So something to do with kinetics, and so there's not much also on the relationship between kinetics and phenotypic variation. Let's say that's also something that's not very explored or known, I would say.
Ok.
Right. Any other questions?
Entendeu.
Ele disse que entendeu aqui. Eu fiquei achando interessante também essa parte da estabilidade cinética, mas o sinal é amplificado, né? We have a sign amplification on kinase cascades. And
yeah, I think it's, again, because of this issue of the, so if you take all the space of possible kinase relationships, most of it is underdetermined. So in fact, this idea of signaling pathways like the ERK pathway or the MAPK pathway, this is really a tiny fraction of what's real. And we also know that if the relationships that are known are extremely skewed by social bias. So the kinases that have the most substrates are the ones that have most publications. So the number of substrates correlates with the number of citations to the kinase. So, the signaling pathways that we know is probably like 2 to 5% of the real thing. So then it's actually quite hard to understand. I guess you could make theoretical models, kinetic theoretical models, and explore those in the computer. But for any actual cell, the parameters and the signaling pathways that we know is something like 2% of the total, probably. I don't know, maybe even less than that. So then it's actually quite hard to make any analysis of that.
Any prediction on this? I understand that. So, people, any more questions? Well, so let's thank you again, Dr. Beltrao, for your inspiring presentation, for your time to come here in this online forum and to give a glimpse of your research. And we truly believe that we can see you in the near future after this pandemic here in Brazil to visit us and to see the formations of our bioinformatics grad students.
Obrigado pelo convite. E se puder ser útil em alguma coisa, podem contactar.
Muito obrigado. É muito inspirador para os estudantes ter essas palestras internacionais, né, Pedro? Então, é muito bacana conhecê-lo, ver o seu trabalho, né? É inspirador, né? Seguramente o pessoal saiu inspirado aqui hoje para fazer muitas investigações aqui.
Isso.
Não é?
E se alunos também quiserem saber mais sobre ciência da Inglaterra ou outros países, ou assim, podem também contactar-me diretamente.
Muito bom.
Isso mesmo. Eu tive, como eu disse, eu tô com uma ideia aqui que começou a buzinar aqui, como a gente fala, né? Mas eu vou tentar melhorar um pouquinho, ver umas pequenas análises e depois eu posso até conversar com você, o Pedro, para dar uma, né?
Sim,
de repente,
claro.
para dizer. Mas muito obrigado mesmo pela palestra e repetindo, espero que você venha aqui nos conhecer. Realmente já era um plano antes da pandemia, né, da gente trazer mais pessoas internacionais, né, Miguel? Tanto para o PPG Bioinfo da UFRN, do Rio Grande do Norte, aqui em Natal, ou da UFMG, que é lá em Belo Horizonte. E a gente fica muito feliz E quem sabe no futuro a gente não se vê lá pela Suíça também.
Quem sabe.
Lá.
Obrigado, então. Foi um prazer.
E
Até a próxima.
have a nice weekend.
Thank you. You too. Bye. Bye, bye. Deus.
Obrigado, Pedro. Tchau.
Valeu,
Isso
Miguel.
aí, João Paulo. Valeu.
Um abração.
Abraço. Até o próximo.
Até o próximo.